Hello, in this video we will discuss about the introduction of human embryology development. So the introduction of embryology we will discuss here. So what is the embryology? Embryology is the study of embryogenesis, the formation of new individual from the fertilization to form a embryo and fetus. So this is also known as embryogenesis. It is characterized by the process of cell division and cellular differentiation. So let's begin to understand from the female reproductive system, which that contain a ovary, fallopian tube, fimbriae, as well as the endometrium. And endometrium contain a uterine wall due to the development due to the menstrual cycle. So the ovarian cycle will develop this wall endometrium through the endocrine hormone regulation. So in this way, this is the fallopian tube, and this basically is the primary follicle are present. The a million of primary follicle develop during the early stage of the female. So in this way, after the maturation due to the stimulus can be hormone stimulus, follicle stimulating hormone will stimulate to produce the development to produce the secondary oocyte, and the secondary oocyte will be ovulate and the corpus luteum will left behind. So this is the uterine wall, and this is the basically go into the fallopian tube. The unfertilized ovum contain a zona pellucida. The zona pellucida is the outer covering of the egg. This is the egg. After this, the many thousand sperms will come toward the egg due to the chemotactic movement, and in this way the zygote will be formed after the one sperm fertilization with the one egg, so to form a two diploid pronuclei stage. It means that fertilization will occur and the two cell stage will be formed. After this two cell stage, the further going on, the 16 cell formation is known as morula. Morula is the mass of cell is in the form of compact come together these cells, and the inside the cavity will be formed is known as the blastocyst. So this is the blastocyst formation, which that is basically implant into the endometrium wall. So 58 cell blastocyst formation after 4 days, and 107 days require the cell 4.5 days, which that require to blastocyst formation. So let's begin to understand the initial stage of human embryo development. So let's begin to understand again this process and how it's possible. Let's begin to understand. So the a pollen body will be formed when the fertilization will be occur. The second meiotic division will be occur in the egg cell, and after this egg cell, the pronuclei will be formed as well as polar body in the first day after the fertilization. And in this way, the cleavage formation will lead to second day, and the third day the tight cell junction will be occur. This tight cell junction during the 3 day, and the compaction formation will be occur. And the fourth day the differentiation will be occur. The meaning of differentiation means the cell will be differentiate. The differentiation means the inner cell as well as the outer cell will be formed, and the differentiation is known as the inside is the blastocele cavity will be developed after the 5 day. So the fifth day, the inner cell mass will be occur. Inner cell mass will be go into the side and compact formation, and the blastocele cavity will be formed. And it is basically the outer layer of the cell is known as the trophoblast. So the outermost covering is known as the zona. So after this, the basically is the complete the zona hatching will be occurring in day 6. Sixth day, that zona hatching will be remove the outer covering of the embryo, and in this way here is the day 7 the uterine epithelium will conceive. This is known as implantation. So after this, the uterine epithelium, which that is able to conceive this blastocyst formation, and the blastocyst formation after the conceiving, the cell mass differentiation will be occur in the day 8th. So this is the implantation will be occur in the endometrium wall, the epithelium, uterine epithelium, and in this way the day 7, which that required the implantation. After the implantation, the cell mass differentiation will be occur. Let's begin to understand. So the day 8th, so the a trophoblast will be formed. The outermost covering is known as the trophoblast without the zona, and the epiblast will be formed and hypoblast will be formed, these different cells. And the epiblast basically is uppermost layer is the blastocele formation, while innermost is the yolk sac, primitive yolk sac formation. So after 8 day, the 12 day covering, which that will lead to bilaminar disc formation. This is the, remember, this is the cross section. We are understanding the cross section of the embryo. So in this way, the 12th day basically is the bilaminar disc formation will be occur. The bilaminar disc is come from the epiblast and hypoblast. So the epiblast and hypoblast formation is the bilaminar disc formation. So after this bilaminar disc formation, here is the day 12, the mesoderm formation will be occur. How it's possible? Let's begin to understand. So the basically is the epiblast and hypoblast. Between the epiblast and hypoblast, the mesoderm formation will be occur. So how it's possible? Because the primitive streak formation in the epiblast and the primitive streak will lead to invaginate is known as the invagination. So this invagination will lead to gastrulation formation because the three germinal layer formation is known as the gastrulation. From the blastulation to gastrulation will be occur. Here you can understand the primitive streak formation, which that will lead to invaginate the epiblast cell into the between the hypoblast and epiblast. So in this way the epiblast will become a ectoderm, and in this way the primitive streak will lead to invagination of the cell. By the differentiation will lead to increase the cavity between the epiblast and hypoblast will become to hypoblast endoderm, and the invaginated cell will give the shape, and the changes of the hypoblast will lead to endoderm by the changes of the cell, and the mesoderm formation is the red color showing in the figure. And in this way the ectoderm formation from the epiblast and hypoblast will develop the endoderm, and between the epi and hypo is the or ecto or meso ecto or endo, the mesoderm formation. So in this way here is the amniotic cell, the day 18, the mesoderm spreading. The spreading of the mesoderm will be formed, the red color is showing in the figure, and the yellow color is the yolk sac, and the mesoderm formation here, and the exocoelom formation. This exocoelom will be formed, and in this way the amniotic sac will be developed in the blue color, and the chorion and the placenta will be developed. Within the placenta, the chorion and amnion formation, which that is surrounded by the amniotic sac, amniotic fluid, and on the other hand the amnion, chorion, and allantois. Allantois, the blue color showing in the which that is surrounded by the yolk sac, is the precursor of the umbilical cord. So this is the chorion, and this is the innermost, this is the digestive tract of the 23rd day. Amniotic sac enlargement will be occur, and this the umbilical cord formation through a yolk sac, which that is the modification, and amnion will be formed. This amnion is basically amnion contain a amniotic fluid, which that is a shock absorber and used for the development of the shape of the fetus, etc. So this was the human embryology development, the overview. Let's next video we will discuss about the another. So in the summary, the fertilization to cell division to morula formation, blastulation to blastocyst, and gastrulation to the neurulation to organogenesis to form a fetus, and complete fetus will develop into the from the embryo. So this was the human embryology embryonic development overview. Next video we will discuss about the gastrulation.